Hello, everyone. Welcome to the Biology Week School STEM Club talk. I hope you've all had a lovely Biology Week Wednesday. I'm Dr. Ali Bune, and it's my honour to chair this session. We have some wonderful speakers for you today. I just wondered if you knew that it was actual National Mouldy Cheese Day. Can you believe that? I don't actually like mouldy cheese. I don't know if any of you do. My students today have done some maggot racing as part of Biology Week. So I don't know what you've managed to get up to. I'm sure it was something absolutely wonderful. Now, before I introduce our first speaker, there's a couple of things I just have to tell you. So just a reminder that this event is being recorded and your cameras and your microphones will automatically be turned off for this webinar. If you've got any questions, can you put them in the Q&A box, please? And we will ask the speakers right at the end of everyone's talk all about it. Okay, so let's begin. So we begin with our first speaker. We're very lucky to have Dr. Arun Pandurangan. He's currently a senior research associate in the Heart and Lung Research Institute Department of Medicine at the University of Cambridge. He completed a PhD in computer science, crystallography, and biophysics at the University of Madras. And he focuses now on the bioinformatics of protein sequence, structure, and function. And he worked on the development of new computational tools to predict protein structure and protein drug interactions. He's currently working on studying the impact of mutations in disease, antimicrobial resistance, and drug development. Arun has several honorary teaching and research associateships. He's passionate about public engagement and outreach in order to support pupils from disadvantaged backgrounds, as well as volunteering for charities, providing support for those in need. So without any further ado, Dr. Arun Pandurangan.
Thank you everyone for joining today for the STEM talk. I would like to thank RSB for organizing it. Today, I'm going to discuss about some of my research work related to protein structure modeling and its relation to drug resistance, especially in the area of antimicrobial drug resistance. This is pretty much an interdisciplinary area that combines multiple subjects, and it might be very useful for STEM students, particularly when there is an interaction between multiple disciplines. So the overview of the talk is I would like to go through some of the compositions of the earth and its carbon biomass, just to appreciate where we live and what are our neighbors and how we interact with them. And then we look at some of the scales of the microbes around us to appreciate the dimensions, and that will give us some information about how to deal with them in the future in the case of infections. And then I would talk about a particular class of biological macromolecules called proteins. These molecules do a lot of functions in the cell and also useful for the general functioning of the organism. And this has high relevance to antimicrobial drug resistance related to drug discovery. And then I'll touch upon a particular tool called molecular modeling, which covers multiple aspects of chemistry, computer science, biology, and mathematics to deal with, to understand biological systems. And then I would briefly touch upon the problem of antimicrobial drug resistance and the possible mechanisms that are related to resistance and how to tackle them. And then I would end this talk by posing certain challenges given by this phenomenon of antimicrobial drug resistance. And through that challenges, we could somehow find our way through this problem. And coming to the first slide, where if you see the slide, you can see I'm showing the composition of the earth in terms of its biomass and the weight of the carbon, basically. So if you see, plants occupy a major fraction of the earth biomass, carbon. It's about 82% of the total biomass. And the second winner would be the microbial world of bacteria that contains about 13% of total biomass, and then followed by fungi and archaea. But if you see the animal kingdom at the bottom right corner, so that is only accounting to about 0.4% of the total biomass, and that contains humans, which is quite a minimal fraction of 0.01% of total biomass. And you can see the apex predators, you can see they are quite minimally represented in the total biomass of the Earth. So this gives you some idea about our presence in this ecosystem, and you can appreciate that bacteria is overrepresented, even though most of the bacteria are actually good for our health. But if you look at the pathogenic bacteria, it accounts for several billion tons of carbon compared to only about 0.06 billion tons of carbon in human. So clearly, bacteria, the pathogenic ones, are the winners. They are everywhere around us, and we need to live with them in sort of in the ecosystem. And if you philosophically think about it, they're also part of the system, and they will try to survive. But we have a greater duty to protect ourselves from this pathogenic bacteria. So there is a war between these two organisms that is taking us to the problem of antimicrobial drug resistance. And then you also may want to appreciate the scale of these microbes and see how they appear before you, whether they are visible or not. Probably you might well appreciate it before that. The microbes are really tiny. Some of them you can see by naked eye, but not all of them. You can see it's been various entities, including microbes, human cells, viruses, bacteria, and the atom placed on a scale that ranges from nanometer to centimeters. Just to remind you, 1 nanometer is 1 billionth of a meter, and 1 micrometer is 1 millionth of a meter. So you can see bacteria, this is like pathogenic ones, fall between 1 micrometer to around 10 micrometer. They can be visible through light microscopes, whereas the viruses are in the range of nanometers, and they can't. You need to have special equipments like electron crystallography, electron microscopy or X-ray crystallography to see them. These are all atomic structures in nanoscale, so you need special biophysical instrument to see them. So that is how tiny they are, but they are everywhere around us. So that's something we have to bear in mind. And just to put things in perspective, 4 nanometer viral particle is about 1,000 times smaller than an ant, and which is in fact 1,000 times smaller than a racetrack. If you just line up the ants in tandem, it will just, if you put 1 million of ants in tandem, it will cover the whole circuit. So you can appreciate how tiny the viral particle is in respect to what we see in the real world. And looking at the bacterial aspects, you can see this is a section of a DNA molecule, about 2.5 nanometer in length. And if you see that in relation to bacteria, a typical bacteria of size about 2.5 micrometer, it's 1,000 times smaller than the bacteria. And the bacteria itself is 1,000 times smaller than a small drop of water. So you can see it's quite an invisible entity, but it is still around us. But don't get me wrong, this DNA, if you unwrap a single DNA in the cell, it will come about 2 meters long. But if you unwrap all the DNAs in all of our cell, then you can go around the solar system twice. So I'm just showing this in terms of a small section to compare in relation to bacteria here. And then, in this slide, what I would like to say is that you can see different type of organisms around us, but there is a basic entity in it that is present in all organisms in terms of biological macromolecules. One of the particular class of biological macromolecule is the protein. Of course, there are other macromolecules like lipids, nucleic acids, and carbohydrates also there, but I'm not going to discuss that because I'm going to talk more about drug resistance, antimicrobial resistance. So proteins are more involved in this process. Of course, other biomolecules are also involved, but our focus here would be on protein biomolecules. So if you see, probably you would have studied in your GCSEs and A levels this very classical central dogma of molecular biology that defines a clear route from a genetic material that you can see in this slide. The genetic material would be in the cell inside the nucleus, as shown here. And this genetic material is in the form of a nucleic acid macromolecule called DNA, and that gets translated into a messenger molecule, which is again a macromolecule called messenger and RNA. And then there are other complexes in terms of proteins. For instance, a ribosome and transfer RNAs come together to decode this code, which is in the form of genetic material, to produce a different macromolecule called proteins, and that will do various other functions in our body. So that is the central dogma that will transfer information from the genetic code to a protein molecule that does some other functions. So this is typically shown in this sort of dynamic picture where the ribosome reads the mRNA segment, and then as it reads, sorry, I have to go back and forth. It will produce a protein, polypeptide chain, as is shown, it's growing here. That protein will do various other functions that is needed by the cell at any particular time. So this is dynamically getting decoded by the ribosome shown in this yellow entity. So that's how the central dogma works. And you know proteins are produced after reading the genetic material. And they're produced for many reasons in our body that we will see in the next slides. But before that, I just wanted to talk a bit about protein molecules in some detail. As you know, proteins are made up of amino acids, and these amino acids there are about 20 amino acids, and they can be classified into multiple groups based on the charge, the polarity, or the hydrophobicity, or there are some special cases of these amino acids. And these amino acids, the 20 of them, combine in multiple ways, forming different lengths of a primary sequence of the protein that will take different conformations that I'll discuss later. So based on these amino acids, you can imagine a primary structure of the protein as something like an example case of a triglycine polypeptide peptide. You can see this is a primary structure, as we call it, and its corresponding atomic definitions are shown here. It's basically protein or made up of amino acids, and these amino acids are made up of individual atoms coming from carbon, nitrogen, oxygen in different conformations based on the side chains that it has. And of course, these protein molecules are highly flexible. As you can see, the flexibility are defined by these conformational angles. You can see, sorry, unfortunately, this is not playing the video. It has to be, what I'm trying to show here is that the protein molecule can actually be flexible around certain angles. That flexibility will provide them an opportunity to form higher order conformational states, which is shown here. As you can see, the protein molecules made up of multiple amino acids, and due to the flexibility, they can fold back and forth, forming a secondary structural element called beta sheet, or they can fold around a cylindrical axis, forming a different secondary structural element called alpha helix. And these are the two basic secondary structural elements a protein can adopt. And these in turn come together to form what we call a tertiary structure, as shown in this picture. In this case, I'm showing a protein called myoglobin, which is the first protein to be solved. And it's made up of 153 amino acids. And as you can see, it contains 8 alpha helix colored differently. One section is shown here. And the main function of this protein is to store oxygen, and it's found in the muscles. So this is one of the function a protein can perform among many other functions, and that we will see in the next few slides, some of them, but we can't exhaustively look at all of them. But still, researchers are puzzled about protein function. They're trying to understand protein more as they go. And these tertiary structures can come together to form what we call a quaternary structure. In this case, you can see 4 tertiary structures come together in a transient, in a non-bonded interaction to form a quaternary structure that will perform a specific function. In this case, it's the molecule of hemoglobin. It will transport oxygen to the body. It carries oxygen and transports to the tissues from the lungs. So as you can see, the proteins can do multiple functions, and a few of them we can show here. You can see proteins can protect us from infection in the form of antibodies, which is present in our body, and these antibodies will recognize antigens, foreign objects, and as soon as they see something new that is foreign to the body, they will recognize this one and neutralize it. That's one of its function, and you can see proteins can also act as a messenger, like in this case, insulin hormone, which will control blood sugar level. So there are other proteins, for instance, enzyme, in this case, amylase, which is responsible for breaking down starch that we eat into fine subunits, fine units that can be readily absorbed by the body for energy. So you can see a small section of a sugar molecule, carbohydrate bound to this protein, which it will break down into small units. That's one of the tasks that this enzyme does. And protein can also be used to copy DNA material, the genetic code that I mentioned before. This is done by DNA polymerase, where you can see a small section of nucleic acid shown in this green and red kind of rose color bound to the protein. So I just wanted to mention these proteins are shown as a different representation, not like atom representation, but as if you roll an atom around an atom to form a molecular surface. That's what it is. These are all shown as a molecular surfaces for abstractions. If you look at atom details, it's difficult to see things clearly. That's why it's shown like that. And then here, there is one more case of a protein doing a structural function, where in this case, it's a microtubule formed by dimers of tubulin protein molecules. Two protein molecules shown here, they're stacked side by side to form a cylindrical structure, which will actually form kind of a railway tracks in the cytoplasm, if you can imagine. And these tracks in the cytoplasm formed by these microtubules can be used to transport cargoes, and also it provides structural stability for the cell itself. And finally, you can see a protein is also involved in transmission of nerve signals. In this case, I'm showing a special protein called membrane proteins. These proteins are embedded in the cell membrane. This is the interface. This is outside of the cell. I mean, this is the inside of the cell and outside of the cell. It's just buried in the membrane of the cell. And this protein the function of the protein is to pump out sodium outside, and the potassium, it will take potassium into the cell, thereby maintaining potential energy difference between the outside and inside of the cell. These energy differences can be seen as an electrical difference or a concentration difference, and these differences responsible for the nerve cells to communicate each other. So these are all tiny molecules doing small, tiny stuffs, but if you put together as an organism, as a human, we do complex functions. So it's good to appreciate what tiny things can do and how difficult they are to understand. And researchers are still trying to unravel the secrets of proteins as we speak. So the molecular modeling will actually try to, what it does, it's try to find the structure and function relationships. Here you can see the structure of a roundabout defines a function that it needs to streamline the traffic. Similar way, proteins also have structure and it has multiple functions, and the molecular modeling will try to answer this problem and using theoretical, computational, and molecular mechanics techniques, and ultimately used for disease and health. And antimicrobial drug resistance is still a big problem. As you can see, the death rates is pretty much higher for higher ages in recent times. So this is big because of lack of antibiotics being produced recently, and this is a problem. And the antimicrobial drug resistance can be done by the bacteria by multiple fashion, like you can modify the drug that is being taken, it can metabolize the drug, or it can modify the target. This case is a drug resistance case, and this case is what we would like to achieve, where the drug actually targets and does it work. And here I'm showing a gram-negative bacteria. And the challenges in AMR are multiple folds, as you can see: access treatments, antibiotic usage, research and development, surveillance, economic impact, international collaboration, and environmental impact. These are multifaceted problems. I believe it can be tackled using artificial intelligence and machine learning in the coming age. So as a STEM student, you don't have to worry about what subject you pick. You can still do great things in the combined fashion. Thank you very much for listening to me, and any questions I can answer later. Thank you.
Thank you, Arun. That was fantastic. It's really good to see our protein knowledge being tied in with something so important as antimicrobial resistance. Absolutely fascinating. Thank you. For our next speaker, we're lucky enough to have Miranda Lowe. Miranda Lowe is a principal curator and marine invertebrate specialist at the Natural History Museum. She was awarded a place on Radio 4's BBC Women's Hour Power List 2020. And she is making a significant positive contribution to the environment and the sustainability of our planet. Her work links science, art, nature to aid the public understanding of the natural world. She's also a co-founding member of the Museum Detox, which is an organisation supporting people of colour who work in an environmental and heritage sector and museum professionals. She works tirelessly on issues concerning representation and inclusion in science, museums, and gallery spaces. So over to you, Miranda.
My talk today is coral reefs, the rainforests of the sea, and the role of the museums, or the museum's collections, in understanding them. So the overview of my talk, I'm going to take you through to understand what coral reefs are, what corals are themselves, the different types of corals, and then how, in terms of my work and my role at the museum, how we actually use a lot of the historical collection of coral skeletons, essentially, to inform current practice in conservation, marine protected areas, and other use of environmental data the museum holds to actually look at and conserve the living reefs. So generally, this is my life in the lab, and I'm currently talking to you from my lab as well with my microscope. So this is what I do on a day-to-day basis behind the scenes at the museum. So the museum has lots of different laboratories and facilities to actually study a lot of flora and fauna from around the world, because we have some amazing collections, predominantly collected originally by the Victorians, but we work with a lot of oceanographic centres and universities to study this material. And this is what it looks like in terms of the coral specimens that I'm responsible for. So what you saw in the first picture was me looking at some lobsters. I'll just go back again. And they're all preserved in a fluid that is 80% alcohol or ethanol, which preserves specimens for hundreds of years, so that we as scientists and actually artists and everybody else, we can examine them, dissect them, extract DNA, all those sorts of things. But most of the coral collections are kept, as we call them, dry preserved. So the coral skeletons, which I'll show you a few examples of. So this is me literally in a big warehouse that is away from the museum, probably about 5 miles away. In another area in South London. So it's away from South Kensington, the main hub of all our specimen collections. But I'm often found in there doing my scientific research and also documenting the various specimens. So what is a coral reef? You can see a beautiful, and I must say that it's a slightly enhanced, very colourful image of a coral reef, but they are colourful in general. So coral reefs are literally homes or hotels for a lot of other animals and other organisms. So the term coral reef refers to a number of different animals that make up part of the reef, which I'm going to explain in a minute. But these include hard corals, sea fans, sea feathers, sea pens, organ pipe coral, black coral, and soft corals. So we're going to take a look at those and what do they mean individually. So literally, you have a lot of different types of corals that make up one big reef. And corals themselves are part of what we call a phylum, and that is Cnidaria. So even though it's spelt with a C, the C is silent. So it's Cnidaria, its pronunciation. And within that phylum, there are lots of other animals. Let's say the cousins of corals are things like jellyfish, hydrae, and sea anemones. And you can see I've shown you 3 examples of different types of jellies or jellyfish. So on your left, a slightly colourful one in a glass jar is known as a siphonophore, and that is a jelly organism that's made up of lots of individuals. And then the one in the centre is a jellyfish that you can find around the coast of the UK, which is a compass jellyfish. And then the one on the right is an example of the Portuguese man of war. And all of these jellies here have varying levels of sting. So the one in the center is being held by somebody, and that has, well, no sting that would affect us as human beings. But Portuguese man of war that's on the right-hand side has a very stronger sting. And there are box jellyfish called cubozoans, and they have a deadly sting. So coral reefs, as I said earlier, are homes to many other animals, and that's why we actually should really care and think about what we are doing as human beings, the way we live, the food that we source in terms of overfishing and things like that, how it might affect the reefs, because they're homes to other animals. So animals such as shrimps and crabs, and other crustaceans, sponges, sea anemones, as I mentioned earlier, sea stars, octopus and squid, so all the mollusks and the nudibranchs or the sea slugs, again, they are very colourful organisms in the world's oceans. And also reefs sustain fishes too, so for schooling and a lot of solitary fish, and also for adult fish to rear the baby fish, so they're young as well. And the reefs are also used by a lot of other animals to escape predation, so to prevent other animals actually capturing them as prey and eating them as well there. And there are certain species of animals that are reef specific or coral specific, so that's why it's really important to have quite diverse reefs around the world existing, because otherwise we won't have a lot of these other animals forming this one big ecosystem, because they all rely on one another. And so if you knock out one out of the chain, one is lost environmentally or due to climate change, then it impacts on other parts of our ecosystem. And I must say, you know, I mentioned that there are shrimps, a type of crustacean that live on the reef, and they're really important because there is a cleaner shrimp that actually, if there is a disease called white band disease that is about to progress across the reef and literally ravage and you know, basically take away all of the soft outer coating and expose the skeleton and causing reef die-off. So that disease can be cleaned up if you have the population density of these cleaner shrimps, because they will nibble away and eat, you know, in terms of that disease, if they're in a large enough population before that disease ravages the reef. So it's really important to have diversity of animals across and living in the reefs. What you may have been aware of seeing, so what I look at a lot, and as I say, we're going to look at them a little bit more closely, I look at the skeletons. So when a coral has actually died, it exposes its skeletons. But when they're living, they have these, and you can see in this image here, these extensions across the skeleton known as polyps. So corals belong to a class. I mentioned the phylum, first of all, Cnidaria, and then we've got a subgroup that is a class of Anthrozoa. And so across a coral skeleton's body, in effect, are there's hundreds and thousands of mini mouths, feeding organisms that are flower-like structures called polyps. And they essentially are like mini sea anemones that kind of wave about and actually capture or sometimes parasitize small larvae and digest. So it helps you know, feed the reef. As I've got here, that they have a simple body plan, which the food passes in and out of a stomach-like sac through a single opening. So those are polyps, and those die off, unfortunately, when a reef dies as well. So you don't get those actually preserved in the dry collection. So here we go into the different forms of corals. So we have things like massive corals or boulder corals. So you have different types of corals that form a reef. So it's like for the boulder corals, such as this brain coral, you can see an example in front of you, they form the basis of a reef, like the foundations of a house here. And many corals have algae called zooanthellae within their tissue. So within this kind of brain-like structure in the middle, you might be able to see on the close-up a kind of darker area. So that's the dried organic matter, and that will be sometimes of the algae. And the algae form a symbiotic relationship with the coral, producing organic compounds that the coral polyps, when it's living, can actually use. So this food source allows the corals to grow faster than they would if they didn't have the zooanthellae. And the algae actually gives some of the corals their colour and help capture the energy from the sun by photosynthesis. So next we have solitary corals. And so these are corals that actually live on their own, and this has a nickname also as a boomerang coral. Fungia is its generic name, and you can see there how it had shaped and it's bent like a boomerang, and that would often be laying on the seabed, so on the lower part of the reef. And next we have plate corals, and kind of all of these common names actually are relevant to what they actually look like visually. So we attach those common names as well as scientific names that we all use, taxonomic names that within the scientific community that we all understand it's the same species that we're talking about. But this is a very thin plate-like coral and would form the bases interspersed at different levels, but would provide platforms for other animals to actually rest on or to actually hide from other predators. And we have a really exciting one here. So when most corals die, they have that bleached look, white and cream. But for some corals, so this is an octocoral. So the polyps that I was talking to you about earlier, so when it's living, they have an 8-fold symmetry. They're polyps, so octave 8, so octocoral, and it's part of the soft coral grouping. So this is an organ pipe coral, and as you can see, when you look at it in more detail, it literally has a lot of these tubes similar to the pipes of an organ. So this is Tubipora musica, and interestingly, this and another coral. So this coral, Tubipora, is red, and when you cut through it, it continues to have red. So it's around, how the structure of the red pigment is embedded in the body or the architecture of the actual animal itself. But there is a blue coral, which I don't have a picture of, called Heliopora Coralia, and that is also bright blue. And when you cut through that skeleton, it has blue throughout it as well. So they're pretty amazing. We have here some examples of specimens that Charles Darwin collected on his 1830 Beagle voyage, the first voyage that he went on. And these specimens are actually displayed within the museum. And interestingly, they were documented within the museum in the 1840s, and they weren't identified to genus or species until 2009. So that just shows how difficult sometimes animals such as corals are to identify, and having the specialists to be able to do that. But in terms of that, we're using our coral specimens, such as Darwin's material and material collected from the Chagos Islands on various expeditions in the 1970s. And this has been, the Chagos Archipelago has been in the news recently because the British government is handing that over to Mauritius. But we have 6,500 corals that are being used in molecular research to understand the living reef, because it became a marine protected area some years ago. So a lot of that environmental data is housed within the museum, so that conservationists and environmental and ecologists can actually use that data for their study. We're also digitizing specimens as well. So a lot of the beautiful images that you've seen, and the speaker before mentioned about scanning electron microscopy to provide the detail. So for those corals that we can't identify with their naked eye, we're using really high level, high powered microscopy to actually provide deeper images. And there you can see that we've been also using some 3D scanning technology as well to reveal mysteries of the oceans, how corals kind of form their reefs, their shapes, and their and how they grow and how fast they grow. So all of this information is there for environmental partnerships that we're forming with other people. And we have a database. So we have the NHM data portal, where you can find out all this information and a lot of the images here to use in scientific research going forward. And then we use a lot of our specimens within and to enable understanding of the public around coral reef in terms of our exhibitions. So this is an example here from 2015. And we actually grew corals throughout the life of the exhibition in a small aquarium. Again, through our lasting impressions gallery as well, we question, what do they tell us about the environment? And this coral that is sectioned here lived in a polluted iron salt factory. And that the red line shows how industry does affect animals such as corals, because you can see the iron through the body of the skeleton of that boulder coral.
Oh, Miranda, that's absolutely fascinating. Are we able to stop you there? Otherwise, we're
yes, we can stop. Yes, exactly. So that was the last slide about what it can tell us about history and who collected. But I will stop there. And if anybody has any questions later on, I'm happy to answer them.
That's wonderful, Miranda. Thank you so much. And just listening to all of that, it made me realize just, how wonderful it is, the diverse biology careers that you can get. It's just fantastic, isn't it? And those lovely images. So our last speaker today, we're very lucky to have Dr. Peter Fretwell. He is an award-winning cartographer, author, and leading scientist at the British Antarctic Survey. He's pioneered the use of satellite imagery to find and monitor polar wildlife, a project that's led him to discover over half the world's emperor penguin colonies, and he produced the first robust estimate of their population. Peter also chairs a scientific committee on Antarctic research's BedMap 3 group and is sensing animal populations from space and heads the British Antarctic Survey's Wildlife from Space group. He's published over 100 scientific papers. He's internationally recognised for his work on studying penguins and other polar wildlife by satellite. He's completed 6 field seasons in Antarctica, and in research stations, ships and in tents. So over to you, Peter.
Thank you very much, Ali. I'll just share my screen. So, yes, thank you very much. I'm Dr. Peter Fretwell from British Antarctic Survey. And as Ali said, I head up the Wildlife from Space group here at the British Antarctic Survey, where we use satellites to detect, count, and study wildlife. So basically monitoring wildlife from 600 or 700 kilometers above our heads using satellites. Now, satellite technology has advanced greatly in the last decade or two. The resolution has got better. There have been more satellites up there. So we can do more and more with this technology. Nowadays, we can get images on the ground which have pixels about the same size as an A4 sheet of paper, 30 by 30 centimeters, which means any large animal, theoretically, we can see, as long as it's out in the open in as good contrast with its environment. The other beauty of satellites, of course, is that they can give us remote access to all over the globe. We can go to the polar regions, we can go to deserts, we can go out into the middle of the ocean, and we can get repeat coverage. We can get multiple images per day of anywhere in the world, as long as it's not cloudy. And even with clouds, there are some radar satellites which can see through the clouds too. You have no bureaucracy with a satellite. You don't have to fill out a paperwork to go and fly a drone or fly your aeroplane over somewhere. You can go into war zones or into really difficult places where you wouldn't be able to get to normally. They don't disturb the animals either. If you try and do a whale survey, for instance, with a ship, often the whales can hear the ship and they'll scarp quite often, and you won't get an accurate estimate of the populations because the actual wildlife will avoid the thing that you're using to try and find them. And overall, they're cheaper than aerial or ship surveys, especially if you want to do large areas in remote places. So just a little bit of lighten the mood. I'm going to just give you a quick, sort of a bit of a quiz here. I'm going to show you some images of animals, and I want you, you can have a think about this and think about what they might be, and I'll tell you. So just a few seconds on each one. So here we've got a sort of bluey green background with some white specks on it. Have a think, what could these be? Lots of them around. These are actually grey whales in Baja, California. Whales are quite difficult to study because they often breed in very low densities, but here they're in quite high densities in a lagoon in the tropics. These are all images of animals we have or are studying at the moment. Here's another one. We've got a white background with lots of brown on it and some little black specks in amongst the brown. I think these are penguins, emperor penguins in this case, in Antarctica. The brown is the guano stain, their poo, which they deposit around the colony because they stay on the ice for about 8 months of the year. Here we've got some little white dots on a green background. As I said, the animals we can look at are anything over about half a metre or so would be applicable. These are actually individual wandering albatross on a French subantarctic island in the South Indian Ocean. These animals have got about a metre in length, each of their bodies, but their wingspan is about 11 metres, but we actually see their bodies. And we can use things like AI to be able to count these automatically, which I'll come on to later. Here we've got another image, white. We're doing a lot of work here. This is the crab-eater seal. The crab-eater seal is thought to be the most abundant, large, wild animal on Earth. But it lives on the pack ice around Antarctica, which is about the size of Europe, changes and is very dynamic, moving around all the time. So actually getting an estimate, our estimates have varied between about 4 million to 60 million because it's so hard to monitor. So we're using satellites over huge swathes of Antarctica now to automate the finding of these animals and try and give us a more robust estimate. Still a bit of work to do before we get there, but I think we will be able to crack that soon. Here, again, we're looking in the sea. We've got a green sea here. This is actually up in the Arctic, in the Russian Arctic, and these are beluga whales. Last year, we published a paper where we discovered a whole population, a new population that had never been known about before of beluga whales, numbering several thousand and quite a rare animal as well. So it's really nice to be able to do that. This is something we've worked on. We've never published on these. You can see lots and lots of little white dots on a green background. Again, this green isn't grass and it isn't sea. This is a lake in Chile. And these are actually flamingos. Now, you can understand how difficult this will be to count manually and how long it would take. So I'm going to talk about some of the ways that we're automating the process, or sometimes we're using other people to count for us. I'll come on to that in a moment. Two more. Here we've got a very desert, dusty environment. You might be able to get an idea of what we're looking at because you can see some cars and vans there. These are African elephants in South Africa in the Kruger National Park. Again, an animal that there's a lot conservation efforts to try and protect them because of the smuggling of their ivory. And we can count the numbers and monitor their populations from space now. And finally, another one that we're doing a lot of work with. This again is in the Arctic. These are walrus, Atlantic walrus off Nova Zelanda, really high in the Arctic, very remote. And it just shows we do a lot of work with those really remote places where you can't get to normally. It would be really expensive or difficult to do. Now, my career, I started using satellite imagery many years ago, and I've been using satellite imagery for a while. My career looking at wildlife started about 15 years ago when, by accident, I discovered that we could see emperor penguins in Antarctica by looking for those brown poo stains on the ice. At the time, we were using freely available Landsat imagery, which has a resolution on the ground of about 30 meters. So each pixel is about the size of a tennis court. So you can't see the individual penguins, but you can see the stains that they leave behind. Now, by doing this and by using higher resolution, freely available satellite imagery since then, we've been able to find all of the emperor penguins. In 2009, we only knew of about 26 colonies, and our knowledge of the distribution wasn't very good. 26 colonies we knew about. We had about 10 sites where we'd seen penguins, but we didn't know the exact location of the colony. Now, using satellites, we've been able to refine that, and we've been able to find over half of all the emperor penguin colonies. We now know there are 66 emperor penguin colonies. We track them and we count them every year using satellites. And this is really important because emperor penguins are suffering in many parts of Antarctica with climate change as the sea ice starts to retreat around the continent. We're tracking their populations and seeing how they're coping and seeing how they're having to move as well. Many of those locations are moving to more stable ice as the ice under them breaks up. Here's an example of the improvement we've got between the Landsat, which we started using in 2009. On the left, you can see, you can just use a pointer here, this brown stain is all we could see, to the 30 centimetre World View 3 imagery we're using now, where you can see each individual emperor penguin. And that allows us not just to see the guano, but to actually automatically find the penguins themselves. And we do that by using what's called an automated classification index. So what we do is we tell the computer what a pixel of guano looks like, what a pixel of ice looks like, what a pixel of shadow looks like, and what a pixel of penguins looks like. And then the computer goes away and classifies, breaks down the image into its component parts, which means that we can then get an area estimate for the penguins. And from that, we can derive the population. Before we can derive that population, we need to know what the area, how many penguins per pixel we've got, which means we need really accurate counts on it. So usually what we have to do then is we have to go and get really good data from the field where we can actually identify each individual penguin properly. And to do this, we have to do field work. We go into the Antarctic or to other areas. And in this case This is me in the Antarctic last November, flying a small drone over emperor penguins to get an accurate count, to calibrate those satellite counts that we've got and understand how accurate our counts are and how many penguins we get in each pixel on a satellite image.
Peter, we've got a couple of minutes left, okay?
Yeah, so you can also go online. We got a project looking at becoming walrus detectives. There's a QR code here. We can give that out later, where you can go on and you can help us and the WWF count walrus from space. We all again did field work there. Here's an example of walrus in the field and the satellites and the drone imagery taken within 15 minutes of each other. So really good there. That's me actually on the right-hand side. You can just see the walrus here, and that's a selfie from space. One of those little dots there is me. But we use a lot of AI to count. Often we've got thousands of things to count, and we use AI as well as citizen science. We often use convoluted neural networks. Again, the technology has been rapidly increasing here, and we're using different types of convoluted network to try and automate the process. You have to train these networks first, telling them what this is. This is albatross in South Georgia. So it goes away and it automates the process, and it tells you how many you've got automatically. We're also doing a lot of work on whales from space. Probably not enough time here to talk about whales from space. But what I will say is that as more satellites are launched, there's great potential to use this technology to count, detect and monitor biodiversity around the globe. And as we know, there is a real biodiversity crisis at the moment, with habitat loss, climate change and pollution driving populations down. So there's an ever greater need to monitor and understand how that biodiversity is coping with the drivers, the anthropogenic drivers that are forcing those populations at the moment. So thank you very much, and I'm happy to take questions with everybody later on.
Oh, thank you so much, Peter. That was wonderful. Isn't it brilliant how you can see all these different places where you could actually work when you study biology? Amazing. Thank you. I would like to thank all three of our speakers. They have been fantastic. If you could all turn your videos on, please, ready for questions. But I think we ought to give them a little virtual clap because they have been brilliant. So a little virtual clap for all three of you. Thank you. Right, we have quite a few questions. So if we could be lovely and succinct in our answer, if possible. And I'm going to start off with Miranda. Miranda, we have a question here saying, how do corals photosynthesize if they're considered animals?
Right, so it's the algae that are photosynthesizing, and combined with the chemical compounds coming from the polyps, so the feeding parts of the coral that provide the coral colour. But it's the algae that are photosynthesizing. Lovely,
thank you. Peter, there's one for you. We have, will high-resolution satellites have a high carbon footprint?
Well, we use satellites that are already up there. So we don't launch our satellites specifically for studying wildlife. A lot of them are used for commercial purposes. We're just sort of tagging on to that. So theoretically, there's very little carbon footprint from our, except for maybe the actual processing from the high performance computers that the CNNs usually have to take. So technically not hard. No, in comparison to, say, a ship, which has a huge carbon footprint, or a plane, which has quite a large one as well.
Lovely topical question there as well, isn't it? Arun, one for you. We have, are novel bacteriophages a way to combat antimicrobial resistance?
Yes, that's an important field still under development, because it was overtaken by development of antibiotics in the 20th century, mainly because antibiotics are a broad spectrum ones. They can attack multiple bacteria in one pill, whereas the phages are already bacteria strain specific. But it's difficult to identify them. But recent metagenomics projects are really coming very helpful to extract phages that are specifically killing certain strains of bacteria. So hopefully it will also be helping us in the near future.
Brilliant, thank you. I think this is a question that probably goes to all of you. So I don't know if you wanted to start off with this one, perhaps, Peter. So it was for all of you. Did you love science at school, and did you always want to be a scientist? So Peter, what do you have to say about that?
I'd say yes, I did. My science career happened quite late in life, but I really, once I'd taken my degree, I sort of really wanted to be a scientist. Didn't get into it till much later, but yeah, I was passionate about science from an early stage, I think.
Miranda?
Yes, I always wanted to be a scientist or have a job in the science world. Originally, when I was doing my A-levels, a pharmacist. I really loved chemistry. Didn't quite get the grades. I didn't fail, but I then moved on to the biological side of things and took an applied biology degree. So, yeah.
Lovely. Arun?
Hi, that's a very good question, actually. Two things define your trajectory, I mean, career trajectory. One is your passion, and the second one is the opportunity. So passion comes first, that you can drive yourself, but you have to seek the opportunities that is not visible, but it's everywhere. If you can do those two things, you can achieve whatever you want.
Lovely. Thank you.
That's what I was inspired, actually.
Peter, I have a question for you here. How deep underwater could your satellites image?
It very much depends upon the turbidity of the water, how much silt and other material is, but also the contrast between what you're looking at and what the colour of the water is. So, for instance, beluga whale, which is white, we can see them quite deep in the water. But a humpback whale, which is black, if you look in the Arctic, which or somewhere that's got quite dark water, not very deep at all. So it really is quite difficult to say, and we have to really think about like how our population that we can how our counts we can see on the from the satellites can be transferred to real populations based upon things like how much time the whales or other things to spend at the surface.
Oh, lovely. Thank you. Miranda, I think this might be our last question. It's for you. What are the 4 major threats to coral reefs?
I mentioned one threat was white band disease ravaging the reefs, and the cleaner shrimps, they need to do their work and be in such high numbers to prevent that. Ocean acidification, the rising temperatures as well, overfishing too, which will impact on the reefs as well, because I mentioned, about various animals are specific to various types of reefs and so on and so forth. I mean, reefs themselves are worth billions to the worldwide economy, and so if we don't have coral reefs, then we won't have certain types of fish as a food resource, or those local communities that live on those islands that have particular reefs as well. So that's a few, but there's a lot going on, and biggest threat is us as human beings, our impact there.
Sadly, yes. So I feel a bit like Columbo now. Not many people are going to know who Columbo is, but I'm going to say just one more thing. So we have one last question for all of you. If you could just pick one thing, it is, what is the favourite part of your job? So should we start off with you then, Miranda?
Favorite part of my job. Gosh, that's difficult. Right. Whenever I open a cupboard and I see something new, or when I think I've seen it all, and I think, oh my gosh, that really existed in the ocean. This is amazing. And also being responsible for anything related to Charles Darwin and his world. I
can imagine. Peter?
Yeah, trickier. The fieldwork's great. But really, I think what really drives me is the discovery aspect of it, discovering new stuff. That's really what drives a scientist. I think it's that thing of doing the new, finding new things, publishing it and showing the world.
Brilliant. Arun?
Yeah, I resonate with Peter, discovering aspects. But then the other one is the collaborative aspects. You meet people and you get ideas from them. That's how things work in the world. You have to collaborate to do big things. So that's encouragement for me every day when I see new scientists or just chat with them over a coffee. Some idea will come over and you just solve a big problem.
Brilliant. That's wonderful. Well, we've come to the end of our session now. So I would just like to again thank all 3 of our speakers. It was wonderful. So thank you. And I would like to thank all of you for attending and all your wonderful questions. And thank you very much. Thank you.